All right, so now let's jump into inductive versus deductive reasoning, which you may have heard about or learned about in previous courses, maybe not even science related, but maybe philosophy or critical thinking class. Um, to break them down kind of simply, inductive reasoning has this tendency to draw general conclusions from many observations. So you're moving from specific to general. Your specific life experience, and you're taking your specific, and this is just an example, but you're taking your specific life experience, so all of your experiences that you've ever experienced, to draw general conclusions about the universe. So let's just say, like all the windows you've ever seen have been square. And so you're going to draw this general conclusion about the rest of the world that, well, all of the windows that I've seen have been square, therefore, all of the windows on Earth must be square. So you're moving from specific, your small window of experience, and you're generalizing that data. You're generalizing your conclusion, or you're drawing a general conclusion. So the reason why it's inherently uncertain is because we're using our small windows of experience. Now, deductive reasoning is kind of the inverse in that it moves from general to specific. So it comes up with ways to test a hypothesis. So here, you start with a theory, so a general rule or theory, something that's well known, and you're taking that well known thing, that global universal truth, and then you apply that to something specific. So, like for example, as a general rule of thumb, we know that gravity acts on things with mass, right? So, as a general knowledge, we know that gravity acts on things with mass. And so, if I were to take this pen and say, okay, so I know that gravity acts on things with mass, I know that this pen has mass, therefore, gravity will act on this pen. And so, I could drop the pen and you could prove, or you could accept your hypothesis in that case. So that's an example of deductive reasoning. Now, inductive reasoning, again, that's the inverse. You start with data. Data could be your own. Data could be from something else. But what you're going to do is you're going to draw conclusions about the broader world from some small amount of data. All right, so here's another example of inductive reasoning. All of the swans we've seen are white, so that's the observation that I have. And then therefore I can conclude that we expect all swans to be white in the world. That's the conclusion. So that's an example of inductive reasoning. So deductive reasoning is kind of the inverse. All humans are mortal. So it's a general rule, generally known thing. Donald Trump is human, therefore Donald Trump is mortal. So the conclusion is true if the premises are true. All right. So let's talk about the scientific method. This one you've definitely learned probably in elementary school or at least high school. So the scientific method is going to start off with some kind of observation that you make. So you make some observation, and then using those observations or referencing those observations, you're going to ask questions. Then following those questions, you're going to pose a hypothesis. So a formal hypothesis usually contains an if-then statement. If something, then something else. Then you create an experiment to test your hypothesis. And as you're creating that experiment, you kind of pose predictions for what what might happen within the experiment. And then from that experiment, you collect data. And then you run the statistical tests to test for significance from your experiment, and then you draw conclusions. You either accept or reject your hypothesis in your conclusion. So here is just a flow chart. You ask a question, you do background research, you construct a hypothesis, which is typically formalized as an if-then statement. Then you test that with an experiment, and then hopefully your procedure is working. You analyze the data, draw conclusions, and you either accept or reject your hypothesis. Okay, so this was a study done by Tyrone Hayes at Berkeley. His observation was that amphibian populations are rapidly declining globally. And so he asked questions. What is causing this decline? Is there a fungal disease present? Is increased ultraviolet radiation detrimental to amphibians' reproductive health? Or maybe is it an agricultural chemical? So the first step here, after asking those questions, after doing some background research, you would create a hypothesis, a testable explanation. So all hypotheses are going to be testable and falsifiable. So even if you come to, if you prove your hypothesis, if you reject your hypothesis, that doesn't necessarily mean that the study was bad. You still learned something. You learned that your hypothesis was incorrect. So the study that Tyrone Hayes did at Berkeley, his hypothesis was if agricultural chemicals like atrazine, which is an herbicide, applied during larval development are causing amphibian decline, then tadpoles developing in water with atrazine will have abnormalities in reproductive tissue as adults. So he pretty much just said that, if atrazine is causing this global decline, then if I throw tadpoles into some water with atrazine, they're going to show reproductive abnormalities. And so his prediction was if we expose larval frogs to atrazine, they will develop reproductive abnormalities. So he designed a controlled experiment that could potentially disprove his hypothesis. So let's test this hypothesis with an experiment. We have 3 tank conditions. We have a 0 parts per billion, so this ppb just means parts per billion. So we had, 1 tank with no atrazine in it, okay? Then he had a tank with 0.1 parts per billion of atrazine. And then we have another tank with 25 parts per billion of atrazine. So he threw 10 tadpoles into each tank, and then he waited for them to grow up to be adults, and then he sacrificed them, and then looked at their reproductive tissues. So the 0 parts per billion, if you notice here in parentheses, we have a control, so it says control. This is actually the control group. This is your reference for what would happen if there was no drug involved. This is your reference point for what do normal frogs look like, or hopefully a reference point for what normal frogs, or the way normal frogs would develop. And then here we have an experimental group, which is a small amount of atrazine, so 0.1 parts per billion. And then we have another experimental group, which is 25 parts per billion in another tank. Now the thing is, in order to be a controlled experiment, you need at least 2 experiments. One needs to be a control. So this is actually an experiment. Technically, this is an experiment. So he's just throwing, tadpoles into, some water, some regular water. And then in this case, here's your experimental group. So this is going to be, a tank containing drug, right? Drug and water, for example. And so he could have actually done this experiment with just these 2 groups, one with no atrazine and one with some atrazine. And he could compare them at the end, and then he could probably draw conclusions. But in this case, he had 3 experiments, one which was the control, the negative control, and then he had, one with a small amount of atrazine, and then one with a lot of atrazine. So he was able to show a gradient, potentially, a gradient in effects. So his results was that, through his data analysis, he found that exposure to atrazine during larval development caused abnormalities in the reproductive tissues of adult male frogs. So he actually found that eggs formed in the reproductive tissues of the testes. Eggs formed in the testes of male frogs. So in this controlled experiment, you have the option, once you've analyzed your results, to accept or reject your hypothesis. In this case, he was able to accept his hypothesis, and he concluded that exposure to atrazine at concentrations as low as 0.1 parts per billion induces abnormalities in the male reproductive system of frogs. So that experiment was a controlled study. So now let's do a comparative study. So what a comparative study is going to do is compare unmanipulated data gathered from various sources. So what he's going to look to compare is probably, he's going to take, for example, you could take atrazine, or not atrazine, you're going to take frogs or tadpoles from a natural environment that contains some atrazine, maybe, or not, and you're going to take tadpoles from another environment that may or may not contain atrazine, a lot of atrazine, a little bit of atrazine, a medium amount of atrazine. And then you're going to look at both of the data and you're going to compare. You're going to compare what happened. So you're going to compare unmanipulated data gathered from different sources. So he's going to start off with a prediction of the differences between groups. And so, yeah, he wanted to confirm the controlled experiment indeed was the cause of reproductive abnormalities in nature. So he wants to see this stuff in nature. So his hypothesis, which is less formal, the presence of herbicide atrazine in environmental water correlates with gonadal abnormalities in frog populations. So he took 8 sites. He looked at 8 sites to collect frogs, or you could say tadpoles, based on atrazine sales. Actually, no, I think in this case, yeah, he's just taking frogs in general, based on atrazine sales. So if there was a lot of atrazine sold in the area, generally if it's sold locally, it's going to be used locally, and so if it's used locally, herbicides could wash off into bodies of water, which is pretty much the whole premise of this. In a lot of our bodies of water, we get more than just herbicide runoff. So we have a lot of accumulation of different compounds in our water that is probably impacting the reproductive success of more than just amphibians. So he collected the frogs from the water at each site, and here's his results. So here, this is actually kind of what surprised me. So the experiment was just to analyze the reproductive organs and water samples for levels of atrazine. So the little stars here is the concentration of atrazine, and then the red is what we really want to look at. So these are the different site numbers, 1 through 8. The stars is the concentration of atrazine, and you would expect a positive correlation between your atrazine concentration and then the rate of testicular oogenesis, which is just the formation of eggs in the testes. And in this case, we actually didn't find that. We would have expected a higher rate of testicular oogenesis at site number 6, but it was about 20% of your frogs had your, you could just say gonadal abnormalities. And then number 3 had the highest amount, but it had like a moderate amount of atrazine. So his conclusion was that the reproductive abnormalities exist in frogs from environments in which aqueous atrazine concentration is 0.2 parts per billion or above. The incidence of abnormalities did not appear to be proportional to the atrazine concentration. So this could be, he's pulling the stuff from nature. There's probably other herbicides involved here, and there's cross-contamination, if you think about it. There's a lot more confounds, than a controlled experiment. You're dealing with water that contains more than atrazine, potentially other herbicides. So in this case, this was a comparative study in which his conclusion probably didn't align with his hypothesis that there would be a linear correlation or a correlation between concentration and abnormality rate. All right.